So my topic is about exploring association between host factors and different virulence factors of Enterococcus species with treatment outcome in enterococcal bloodstream infections. And this topic was of my student who is my guiding as a PhD student. And we are working on whole genome sequencing for exploring more genetic genes for AMR as well as for virulence factors. And this is a byproduct of our work in which we got the genetic analysis of our bacterial isolates like Enterococcus and their genetic factors. But as a hospital, we get these bloodstream infections very commonly, and we wanted to find out because enterococcal bacteremia generally occurs in very debilitated patients. So we just wanted to find correlation between the virulence factors and the host factors like comorbidities in outcome of the treatment. So going ahead with the topic, like Enterococcus species, mainly Enterococcus and faecalis and Enterococcus faecium, have evolved over the years, leading the leading cause of variety of nosocomial infections, including bloodstream infections. And most of the BSI have endogenous source. BSI with enterococcus generally occurs in very debilitated patients. In case of patients on long-term antibiotic therapy, enterococci dominate as a, mostly they are very resistant to third generation cephalosporins, and when the intestinal flora is killed, they overgrow and they dominate the gut flora, and because of their ability to tolerate the high levels of antibiotic uh, and their intrinsic resistance. Virulent factors help them facilitate decolonization and evasion of immune system and cause vascular infections. Enterococci are supposed to be good colonizers not only of the intestine but as well as the vascular endothelium also. In this study, we also tried to attempt the probable role of patients' comorbidities and the clinical outcome after treatment of such PSI. So our aims were to study different virulence genes and their functions by WGS, to study the role of different virulence factors in pathogenesis of enterococcal bloodstream infections, and to explore newer strategies. Because at the end of it, I'll explain why we now need to explore the newer strategies because antibiotics will not be of much help. So coming to material methods, during the period of April 2023 to June 2024, we had 99 positive blood cultures. Amongst 300 blood cultures we did, they were all enterococci, and we only chose the MDR, XDR Enterococcus species, and most of them were MDR, XDR to see the genetic makeup of by whole genome sequencing. We identified the isolates by YTEC2, which is an automated system, and confirmed the isolates by MARDI-TOF MS and also by whole genome sequencing. So whole genome sequencing, the study of virulence genes was carried out on 25 isolates of Enterococcus species. We also noted the host comorbidities to find an association between treatment outcome of enterococcal bloodstream infections. We did the ID and susceptibility using YTEC2, and according to the CLSI guidelines, we use the panel of antibiotics like benzyl penicillin, high-level gentamicin, ciprofloxacin, levofloxacin, erythromycin, linezolid, teicoplanin, vancomycin, and tetracycline, digoxin, nitrofuran, and daptomycin as we keep it for the enterococci causing bloodstream infections. MALDI-TOF, like Enteromus France, was used for bacterial identification by a proteomic method, and all the strains under the study were MDR. Whole genome sequencing, the DNA extraction was done by manual method, by CTAB, and DNA quantification was tested using cubic fluorometer. Nextera XT kit Illumina USA was used to prepare libraries, and sequencing was done with Nova Sequence 6000 S2 reagent kit, BL5 300 cycles on Nova sequencing 6000 machine from Illumina USA to obtain 2 into 150 base pair rates and coverage of at least 1000x. The sequence analysis, the quality control and trimming were done by fast QC. Unicycler was used to assemble genomes of every enterococcal isolate. QC of assembly checked by QAST. The published profile was used to determine the multi-local sequence type, MLST, for each isolate. The resistome and the virulome was analyzed using AMR, Finder Plus database. So we had 99 Enterococcus species positive blood cultures, of which Enterococcus faecium was isolated in 72 and Enterococcus faecalis was isolated in 27 out of 99. And then the male-female ratio of BSI, or the male 53 were with E. faecium and 18 were due to E. faecalis, while in females 19 were due to E. faecium and 9 were due to E. faecalis. These are the virulence factors or virulent genes that we detected in all the isolates. We have compared because there is a lot of controversy like things in treatment of Enterococcus faecium and Enterococcus faecalis because their virulence and their AMR, the resistance mechanism is very different and the susceptibility pattern is also different. It is said that most of the Enterococcus faecium are intrinsically resistant to vancomycin. So it is very essential that we identify the species scrupulously before we put the patient on selective treatment. So the virulence factors for adherence like ACM and ACE, the ACM was found in all the isolates of Enterococcus faecium, but it was not there in the faecalis. But ACE, which is another surface protein which is required for adhesion, was found in 5 out of 7 Enterococcus faecalis, was not found in Enterococcus faecium. The endocarditis and biofilm-associated pili, APPA, APPB, and APPC were seen in all the isolates of Enterococcus faecium and 6 out of 7 of Enterococcus faecalis. Biofilm formation genes responsible like ASRTC was found in all Enterococcus faecium, but 5 out of 7 in Enterococcus faecalis. Capsule biosynthesis gene like ECBA was found in 11 out of 18 in faecium and 5 out of 7 in case of faecalis. Adhesion and biofilm formation like EFA was found in all isolates of Enterococcus faecium and 6 isolates out of 7 in Enterococcus faecalis. Surface adhesions like SGRA was found only in Enterococcus faecium, was not there in the Enterococcus faecalis. Pili formation like pill was found in 2 isolates of Enterococcus faecium, but was not found in Enterococcus faecalis. Capsule formation and antiphagocytosis gene virulence factors were found like CPS-As and UPP-As and CPS-B and CDS-A were found in most of the isolates of faecium and almost 86% of Enterococcus faecalis. Biofilm formation like BOPD and FSR were found in all isolates of faecium and almost 71% of faecalis. FSRA was not there in a case of faecium, but was there in 3 out of 7 in case of faecalis. A capsule formation CAP group was found in 6 out of 18 faecium isolates, while they were there in all faecalis. Hyaluronidase, hemolysin were found in only 1 out of 18 in case of faecium and 5 out of fecalis. And the gelatin, cytolacin, and serine protein is found only in fecalis, but they are not found in faecium. These are the chart of comorbidities that we found. We found that the kidney failure or CRF patients were more prone to get BSI, like 36.41%, followed by CNS diseases like acute encephalopathy or cerebrovascular stroke, middle cerebral artery stroke, subdural hematoma and ischemia stroke was 15%. The blood disorders, the digestive disorder like small bowel obstruction, the percentage wise they were very minor comorbidities. And the liver disease, there were 12%, 12 patients who had liver disease, and they suffered from Enterococcus BSI. We also see the malignancy cases. There are 4 malignancy cases, and 4 of them had multiple comorbid like diabetes, mellitus, CRF. So they were also considered in the chronic renal failure due to diabetes mellitus. We also found some newer virulence factors because it is the first kind of study which was done on human isolates about the genes. Earlier studies were done for the genetic virulence factors. They were on animal isolates. So besides the major, and most of them were done by PCR, this is the first of its kind which was done on WGS. And thanks to the Biotechnology Center at Gandhinagar who helped us in the whole genome sequencing. We also, along with the major virulence factors, we also got this newer virulence factors like SGRA, SLRA, RGPG, and we still have to evaluate the role of these virulence genes in the virulence of Enterococcus in bloodstream infections. So this we already talked about that major comorbid conditions were associated with our study was CRF followed by CNS disorders and liver disease, and minor were autoimmune disorders ongoing. The another virulence genes which are responsible for tissue invasion like hyaluronidase, the HYL gene encodes for the hyaluronidase enzyme which helps the bacteria to invade host tissues. The genome analysis by earlier workers explored the genetic diversity of and virulence profiles of Enterococcus isolate from blood cultures. The HYL, GEL-E, and CYL-A, which are known to contribute to immune system evasion and host cell damage. Their findings highlighted the value of these genes in the pathogenicity of Enterococcus bloodstream infections. Our study found that HYL gene was detected amongst Enterococcus fecalis 5 out of 7, while Enterococcus faecium only 1 had this. The GEL-E gene for gelatinous formation is another virulence factor gene that upsurges the ability of Enterococcus species to cause disease. Gelatinase is a matrix metalloproteinase enzyme that hydrolyzes gelatin, collagen, and other proteins. It plays an important role in the development of endocarditis and prevents complement-mediated response. The GEL-E gene is so co-transcribed with GEL-E, SPR-E gene, which encodes a serine protease. Together, these 2 genes contribute to the virulence of Enterococci. Our study also found genes associated with biofilm formation like EFAA and BOPD, which are 96 and 92% in case of fecalis and faecium, respectively. For a better understanding of a bacterial mechanism of causing pathogenesis and its interaction with the host, only one of the fecalis was vancomycin resistant, while uh, most of the Enterococcus faecium were intrinsically resistant to vancomycin. Uh, The research delivers valuable insight into the pathogenesis of multi-drug resistant Enterococcus infections and guides targeted therapeutic strategies which are custom made for the individual, in the sense that because the comorbidities are so much in a patient who eventually gets infected with the enterococcus in a bloodstream infection, you cannot help the comorbidities. What you can tackle is the virulence factors of this organism, which should be the targets of newer therapeutics. And that is the way you can overcome these infections. Our study indicates patients with CRF are at the highest risk of enterococcal BSI followed by CNS. According to the present study, the treatment outcome in patients with enterococcal bloodstream infections, the mortality is very high in patients with CRF, followed by kidney disease and liver disease. The study confirms the presence of various virulence factors genes in clinical enterococcus isolate from bloodstream infected and immunocompromised patients of ICU. The virulence gene study suggests Enterococcus faecalis is more potent pathogen or is a good pathogen having more virulence factors than Ephesium genetically. But what we found was the Ephesium was more commonly associated with bloodstream infections than Enterococcus faecalis. The study suggests about the future studies required to be done on newer treatment strategies involving inhibition of these virulence factors to overcome dissemination of enterococci, so that improving our understanding of the pathogenic mechanism is crucial. And there are studies which have done to counter the virulence factors. More studies are required to be done to prevent enterococcus entering into the blood. Countering the virulence factors would be more helpful in such BSI as they are seen more commonly in immunologically debilitated patients. We must develop strategies to prevent the consequences of these infections while addressing the spread of antibiotic resistance. All the progress has been made, but still there are a lot of changes ahead. These are my references. Thank you.